When we make the conscious choice to take the hard route or expend the effort to go the extra mile, we are relying on a highly specific region of our neuroanatomy. That region is the prefrontal cortex, or PFC. Because it houses our capacity for executive function, neuroanatomists refer to it as the personality center of the brain, the physical location that defines human character. The PFC acts as an integration hub. It takes in moment-to-moment input from our surroundings, compares those variables against past experiences, and translates that data into insight and long-term planning. In 1848, the case of Phineas Gage offered a window into this anatomical reality when an iron tamping rod pierced through his orbitofrontal lobe. While Gage survived, his behavior inverted. A respectable, loving husband and father became short-tempered, brash, and incapable of following through on the plans he devised. Medical observers at the time noted that the physical damage to Gage's frontal lobe coincided with a sudden, total loss of his social inhibitions and executive decision-making. Clinical deficits in the prefrontal cortex leave basic movement and sensation intact, while altering the biological mechanisms that construct a person's identity. The prefrontal cortex functions as a high-level association area, distinguished from the brain's specialized sensory and motor weightins. Looking at this 3D model, we can see the exact boundaries of the prefrontal cortex as defined by standard anatomical mapping. Rather than passively receiving external stimuli, the PFC serves as an active processing system that receives input from multiple distant cortical regions. This diagram illustrates these connections in action. Reciprocal connections allow the PFC to pull in varied data, adapt to situational surroundings, and modulate physical reactions. A critical part of that modulation is the inhibition of primal survival responses originating from the primitive limbic system, allowing us to suppress instinct in favor of social cooperation. Pro-social interaction is dependent on this central hub's capacity to continuously integrate internal biological data against the demands of external social reality. The physical structure of the PFC begins its development in the 3rd week of gestation, originating within a layer of embryonic tissue called the neural plate. By the end of that 3rd week, the neural plate deepens to form the neural groove, which then completely seals itself off from the amniotic fluid. This fused structure becomes the neural tube. In the 4th week, the prosencephalon, the primary vesicle of the forebrain, bulges laterally out of the neural tube. This further differentiates into the telencephalon, which matures into the cerebral cortex. During early development, the PFC experiences a massive overproduction of neurons and synaptic connections while in utero. To function efficiently, the brain initiates synaptic pruning. This pruning eliminates excess connections through early childhood to establish defined neural pathways. Because the PFC is one of the final areas of the brain to fully mature, it remains vulnerable to disruptions during late fetal and childhood development. The adult PFC is the result of precise biological subtraction and refinement. Sustaining this refined network requires constant metabolic resources. The PFC connects to the rest of the nervous system via fasciculi, large bundles of axons that serve as reciprocal data links between cortical areas. Here we see a cross-section of these association bundles. The superior and inferior occipitofrontal fasciculi serve as the primary long-range superhighways for the cortex. The cingulum and uncinate fasciculi link the PFC to the limbic regions and the sensory cortices. These bundles allow the PFC to receive sensory updates and transmit executive instructions across the entire brain simultaneously. The prefrontal cortex acts as a conductor that can only communicate with the rest of the brain through these dense white matter cables. When these physical and chemical pathways deviate from a standard biological baseline, we see the emergence of physiologic variants associated with depression and schizophrenia. This chart visualizes data from an mRNA assay, a laboratory test that measures the expression levels of specific genes. In patients with depression, assays reveal a downregulation of the genes responsible for transporting glutamate, a major excitatory neurotransmitter. In patients with schizophrenia, functional NRI studies show irregularities in network timing, specifically reduced long-interval cortical inhibition and general prefrontal hypoactivity. In extreme cases, the variants are structural. Holoprosencephaly is a developmental condition where the embryonic forebrain fails to divide into two hemispheres, resulting in a single ventricle brain structure and reduced prefrontal volume. Exposure to severe early childhood stressors can also physically alter developmental pruning, creating lasting changes in adult PFC activity. Neuropsychiatric conditions present as the clinical result of a physically malformed or chemically underactive prefrontal network. Clinically, the PFC is divided into two functional zones, the lateral prefrontal cortex and the ventromedial or orbitofrontal prefrontal cortex. This brain map highlights the lateral PFC. Lesions in regions like the dorsolateral PFC typically result in the loss of working memory and an inability to adapt to changing rules. Lesions in the ventromedial PFC cause emotional and judgment-based deficits, leading to poor decision-making and confabulation, the unconscious generation of false memories. Because of the high stakes in frontal lobe neurosurgery, surgeons utilize functional mapping while the patient is awake to test language and motor function in real time. The critical motor and language centers lie strictly posterior to the prefrontal cortex proper, providing a structural buffer zone during operations. The prefrontal cortex manages our identity and social existence. That capability is dependent on a precise arrangement of embryological pruning, redundant vascular supply, and dense fascicular wiring.